Hi, this is Jessica. I'm Masters in Nursing. With my extensive learning and guiding experience of more than 5,000 hours, I have been guiding and consulting university students with their various academic projects. Today, I will be taking you through one of the most common types of projects that academic scholars are generally asked to construct as an integral part of their curriculum. I will guide you through the process of conducting in-depth research. I will lead you through the process of drafting and compiling well-crafted and profile-like write-ups with the assistance of a sample. Let us have a look at the sample for a comprehensive understanding of the subject matter. Today, we are going to discuss about a bioinformatics assignment, that how we are going to make an analysis for a bioinformatics one. Bioinformatics is an emerging field all over the worldwide, that is taking the attention of researchers to discover new proteins, new genes that helps them to understand better about the diseases, about the conditions or genetic disorder that has been taking place and leading to the chronic condition in patients or in among a population. So moving ahead with the sample and its requirement. So the assignment is about novel gene discovery project, which is homology analysis. Homology is about finding out that what is a similarity that has been existing between two sequences that better help to draft a medicine, better help to draft a treatment for a particular condition. So the aim of this exercise is to help you to understand that what is homology analysis and comprehend that how we are going to use BLAST tools. So BLAST is a tool that help us to determine that what is the similarity lying between two sequences. So there are examples of T-BLAST-N, BLAST-X and BLAST-P. These are different types of BLAST that we are going to use for various sequences, whether it is a nucleotide one or whether it is a protein one. And the assessment will include selection of a protein from an organism that is of our choice to discover a novel gene, and then taking for that particular gene for homology and taking out the gene and searching using the BLAST tool. So what we have to do, we have to select one protein. For example, I took a protein from Homo sapiens, that is humans, and I took a protein from any another species, whether it is E. coli or whether it is Drosophila melanogaster and so on, and we are going to put up a novel gene against a present gene, and then we are going to perform BLAST to see that how much similarity is existing. We have to provide the document in the IMRAD format. So taking the IMRAD format, it is about introduction, methodology, research, discussion format. So we have to state our aim and objective, that what is the specific analysis is all about, describing the method that we are going to use, what is the critical justification of that method, what method we are going to use, for example T-BLAST, BLAST-X, BLAST-P, which one we are going to use, describing with the help of table, providing the findings and giving the in-text citations. Now this is the guidance that how we are going to perform. It is helping us to make inclination with our work, that what are the steps that we have to follow while performing BLAST and then interpreting the results. So we have to first name the protein that we are interested in and give the accession number. So for this we have to go on NCBI. NCBI is the database that is a repository for all the sequences, whether it is protein, DNA. You are going to find each and every kind of sequence on NCBI. So we have to retrieve the protein of choice from that. While opening the when we open the NCBI page, we are going to get an accession number, the FASTA sequence and so on. We are going to get an article as well in which the sequence has been described or whole research has been taken. So it is very useful while making any bioinformatics analysis. Then we have to perform BLAST against the DNA databases, for example genomic or DNA ESTs. So likewise it said NCBI or EBI BLAST. So we have to take care of this, that what particular software we are going to use. After retrieving the database, we have to go for BLAST to find out the homologous sequences or homology in this among two that sequences of query protein and the selected one. And then we have to do pairwise alignment, in which we have to take care of e-value and score for inspections. E-value is about the similarity that has been existing between two sequences. More the value, more the sequence similarity will be there, and score is there in the results of BLAST. When you are going to run the results on a BLAST page, you will be seeing the e-score, the alignment score, the percentage, the gap, the identity, so on. So these were about the blast result, then interpreting the blast results, gathering the information about that novel protein, and moving ahead with further information in terms of aligning that information, that how it is different from that, and what we are going to know about this novel gene, and how this will help us to take our research further in any another field. So presenting the work from 1 to 4 with the help of a poster. So this is the IMRD format, that is introduction, method, research, results and discussion. This is about a poster that we have to take care that the information that we are going to drive is gonna present in a poster. So this is about a poster. So let's see that how poster will look like. So this is the poster in which we have provided the background, that what is the background of this. So this is the background, that which protein is selected is insulin and how it is different. Faster format has been taken. Aim and objective has been defined, that what is the sample and what we are homology we are going to find out. Method we have described, that we are going to use which, that is GROES protein is the selected one that we are going to further take for our analysis. Then results. So results is there. This is the results that is describing that what is the gap. This sequence, the gray region is showing the similarities and the red one is showing the gaps that is existing in the sequences, and this one is showing that how much blast hits we are getting from this particular sequences. So this is about the blast, and for presentation in a format, in a IMRT format, that is describing that how we are going to further take the bioinformatic analysis. This is about presenting and doing the bioinformatic analysis. I hope this sample was effective for you and will assist you with your future research projects. In case of any query, please reach back to us. We would be glad to assist you in your academic course and research work. Thank you.